Good morning, everyone, and thank you all for attending today's Journal Club by the ATS Assembly Respiratory Structure and Function Group, moderated by the Early Career Professionals Working Group at the ATS. My name is Niyati Borkar, and I, along with Dr. Thu Tran, will be moderating today's discussion entitled Asthma and Lower Airway Disease, Neuroplasticity, and Neuroimmune Interactions in Fatal Asthma by Carly Koster, who is a PhD student affiliated with the University of Groningen in the Netherlands, working under the mentorship of Dr. Renaud Gozens and Barbara Mergert. Today, Carly will be presenting her recent publication, Neuroplasticity and Neuroimmune Interactions in Fatal Asthma, which was published in Allergy earlier this year. We are also joined by experts in the field today. We have Dr. Matthew Drake from the Oregon Health and Science University. Dr. Drake's research program investigates the role of neuroimmune interactions in the development of airway diseases such as asthma and cough. We are also joined by Dr. Yujwan Su, who is a project scientist in the Department of Pediatrics and is a postdoctoral researcher at UCSD, working with Dr. Zin Sun. Dr. Su's work is centered on the intersection of neurobiology and pulmonary medicine. Lastly, we have Dr. Thomas Taylor Clark from the Department of Molecular Pharmacology and Physiology at the University of South Florida. Dr. Taylor Clark's research is centered on the role of nociceptive sensory nerves in healthy as well as diseased airways. We'll have approximately 25 to 30 minutes after Kali's presentation for discussions by our expert panelists, as well as questions that the audience may have. Please use the Zoom chat function to enter your questions at any time. As a note, this session will be recorded and posted on the ATS website. With this, I'll let Kali begin their presentation and take the stage. Thank you.
Thank you, Niyati, for that introduction. Yes, so welcome everyone. Good morning or afternoon, depending on where you are. I'm Carly. I'm a PhD student at Groningen University, and today I will be presenting a collaborative publication with the University of Sao Paulo that we published in Allergy, beginning of last year, early this year. Let's see. Yes. Yeah, so this is the paper and all the co-authors of the paper that we published in Allergy. So I'll first give a short introduction on asthma and neuroplasticity and related neuroimmune interactions before I dive into the content of the paper specifically. So I think most of you are probably aware, but just in case, a short introduction. Asthma is a disease that affects over 300 million people worldwide. It's characterized by bronchoconstriction and mucus production. Just get this out of the way. We have airway inflammation, and together this results in the narrowing of the small airways, limiting the breathing of the patients. And patients often suffer from coughing, wheezing, chest tightness, and shortness of breath. But recently, there's also been a new pathological feature identified in mainly severe asthmatic patients called neuroplasticity. And a feature of the lungs that people often forget is that it's very densely innervated. So this is a nice video from Scott et al. In which they show PGP positive innervation of the airways of a murine model. And we can clearly see that the airways are very densely innervated. And it's maybe an obvious thought that something that's so present in an organ can also be affected during different diseases. So the lungs have different, the innervation of the lungs can be subdivided into two main types, I would say. The efferent innervation, which is mainly cholinergic and runs from the brain to the airways, mainly inducing muscle contraction. And then we have sensory efferent innervation, which is mainly from the vagal ganglia, and a small percentage comes from the dorsal root ganglia. And they mainly bring information from the airways to the brain, and then they measure things such as muscle contraction, but also other stimuli, chemical stimuli, pH differences, etc. And then I mentioned this pathological feature of neuroplasticity. So in general, neuroplasticity is often defined as the ability of the nervous system to adapt itself to persistent stimuli. And for asthma, we observed this in the lung as an increased neuron density, increased neurite length, and a lowered firing threshold of these nerves, which contributes to the hyper-responsiveness in asthma that these patients have to specific and aspecific stimuli. And this is often linked to different neuroimmune interactions. And then you can think of different immune cells such as macrophages, eosinophils, neutrophils, ILC2s, and mast cells. And this is not only studied in the lungs, but it's also been observed in, for example, the skin or the gut, and contributing there to different other diseases such as eczema or irritable bowel disease. And this communication of these neuroimmune interactions is often thought to be bidirectional. So on the one hand, we have immune cells that produce mediators such as neural growth factors or neurotrophins. And on the other hand, you have neurons that can secrete inflammatory mediators. And then you think of CGRP, substance P, VIP, the vasoactive intestinal peptide. For a long time, there was a bit of controversy how or whether this neuroplasticity, how that actually took place and whether it took place, because different studies said they did observe this, other studies did not. So one of the first studies that really showed concrete that it's only present in persistent asthma is actually from Matthew Drake, who's also present today, which they showed that for persistent asthma, we observed this increased nerve length and branch points, while for intermittent asthma, this is not actually observed, so that you really need these persistent asthma patients in which we see this neuroplasticity. And this was later confirmed in a murine model, which they showed that you also need a chronic overexposure to really induce this PGP neuroplasticity. And they also showed this specifically for cholinergic nerves in human samples, in which they showed increased FACT positive, which is a cholinergic marker in asthma patients compared to healthy controls. So at the beginning or before we worked on this study, we already knew that this neuroplasticity is a pathological feature that is observed in persistent or severe asthma, and that there is this phenomenon of neuroimmune interactions which can communicate in a bidirectional manner. So we wanted to confirm the presence of this neuroplasticity in fetal asthma samples, together with seeing if we can identify co-localization of these nerves and immune cells in human asthma, and investigate the effects of this spatial co-localization on a transcriptional level specifically. So here's an overview of the study that we published. So we had this cohort of 12 fatal asthma patients with no significant differences in age and sex. There was some smoking history, but they all had persistent asthmatic symptoms, and uniquely there was no prescription for oral corticosteroids, and we compared data from this group to 10 control samples. And we observed in this, using immunohistochemistry, we observed an increase in PGP 9.5 area, so a pan-neuronal marker. We observed an increase in CGRP expression and an increase of mast cells, eosinophils, and Canon-1 positive cells in this close proximity to these nerves. And then we studied more specifically how the interaction between mast cells and sensory neurons, what specifically happens there using an in vitro model. And we then performed RNA sequencing on the mast cell population of this model. So recap, we did the immunohistochemistry on the fatal human asthma samples in comparison to a control group. And then additionally, we did an in vitro study to test, to analyze the gene expression profile. Yes. So first we validated, of course, the asthma, the asthma in the fatal human asthma samples. So we looked at eosinophilic inflammation, but also mucus cell hyperplasia, smooth muscle hypertrophy, and thickening of the reticular basement membrane. So this is an example where you can clearly see the asthmatic phenotype in the airways of the fatal human asthma patients compared to the control sample. But of course, we were interested in the innervation. So we looked at the presence of PGP 9.5 positive areas. Here you can see an example of the fatal human asthma, in which you can see in purple the PGP positive area that is increased compared to control PGP positive area around the airways. And additionally, we looked at CGRP expression. CGRP is this sensory neuron marker, and we quantify this. And here you can see that in the fatal human asthma samples, we see an increase of CGRP positive area within the nerve, different nerves in the fatal human asthma samples. So here in the brownish color. And if you then control for the fact that we have an increase in nerve bundles, which we already observed with this PGP 9.5 positive area, then we see that this is really a CGRP increase within the nerve bundle. So in short, we see increased PGP 9.5 area around the airways of the fatal human asthma samples, and then an increase of the CGRP positive area within those nerves. So this indicates increased innervation and either increased CGRP production by the sensory neurons or a change to the nociceptive phenotype of the nerves. So specifically, this CGRP is a neuropeptide which binds to heterodimerized receptors, the COLC receptor in combination with RAMP1 of one of the RAMP receptors, so RAMP1, 2, 3, or 4. And these receptors can be expressed by different immune cells. So this links to the neuroimmune interactions that I introduced earlier. So the next step was to investigate whether we can also identify immune cells in the direct vicinity of the nerves of fatal human asthma patients. So we first looked at mast cells. So here you can see with the tryptase staining in green-blue that there is an increase of mast cells around the nerve bundles. And we quantify this in different areas around the nerve. So directly touching the nerve within a radius of 25 micrometers, 50 micrometers, or all of those added up. And you can see that for all 3 areas around the nerve, we can see an increase of these mast cells. And then we did the same thing for eosinophils. Here with the Congo red staining, we identified eosinophils around airway bundles, nerve bundles in the airway. And we quantified the number of eosinophils directly touching the bundle within a radius of 25 micrometers or 50 micrometers or in total. And it's been previously published that KNM1 is one of the molecules, or KNM1 interaction is one of the ways through which mast cells and neurons can bind to each other and interact, and that they influence also mast cell degranulation, so that this KNM1 dependent contact between sensory nerves and mast cells induces mast cell degranulation. So we also wanted to look at this KNM1 expression. Yes. So here we have identified KNM1 positive cells both within the nerve bundle and again in the vicinity, so touching within a radius of 25 micrometers and 50 micrometers. And we see again this increase of KNM1 positive cells around the nerve bundles. But if you look at the amount of KNM1 within the nerve area, either expressed by the nerve or by cells infiltrating the nerve bundle, we do not see a direct increase. So in short, we see increased mast cells, eosinophils, and KNM1 positive cells in directly touching or within 25 or to 50 micrometers of the nerve bundles, of these PGP nerve bundles. But we see no increased expression of this KNM1 positivity or positive cells in the nerves. And it's good to note that there is other cells besides, for example, mast cells that express KNM1, so it doesn't necessarily indicate the presence or overlap with the presence of mast cells. But still, we were interested in what could be a potential mechanism for these mast cell sensory neuron interactions. So we set up an in vitro model based on human pluripotent stem cell derived sensory neurons. So this is the protocol that we use. We have human pluripotent stem cells that we can differentiate into the vagal neural crest precursor cells. So as I mentioned at the beginning, most of the sensory innervation to the lung comes from the vagus nerve. And from there, we can then differentiate them into a sensory phenotype and then mature the neurons for subsequent co-culture. And here we did this with mast cells, a mast cell line called LUVA cells. So an indication of what this looks like over the 45 to 40 to 45 day protocol. And this is a short, I hope that I can, oh, I think I need to turn off the laser pointer to play it, yes. So this is an incusite video of the differentiation from this vagal neural crest precursor to the eventual sensory phenotype. So this is about 20 to 25 days. So I can click a little bit through it to not have it take too long. So you really see that we already start very early on with the formation of axons or dendrites, like a network, and then this network becomes more and more dense. And then I think at the end, we even can identify some of the mast cells that are added to the co-culture here that interact with these nerves. So this is an overview of the co-culture model. And if I go back to my laser pointer. Yes. So first we confirmed this or the expression or the differentiation also of this markers using immunofluorescence. So here we have identified the neuronal network with beta-3 tubulin, another neuronal marker, and the mast cells with Fc epsilon receptor. And we can see that these mast cells or these LUVA cells really sort of migrate towards the neuronal structures and attach to these HPSC-derived sensory neurons in this co-culture model. And we then separated the two cell types in this co-culture model using max-speed system, separating them on PSA NCAM expression. And then we performed RNA sequencing on the mast cell population. And here you can see a separation of the co-cultured mast cells and the monocultured mast cells. And we then looked into, based on what we already saw in the CADM1 study that I mentioned earlier, that this can induce degranulation. We looked at differentiation and activation markers. And we saw an enrichment of this mast cell differentiation that was upregulated. So we looked at genes such as c-KIT, GETA2, also HTC, and we saw an upregulation of this gene expression profile in the mast cells upon co-culture. And then based on also this CADM1 study, again, we also looked at the expression of these receptors. So first, the expression in the monoculture cells was basically absent the raw counts. So it was too difficult to identify with RNA sequencing, but you do see that for CADM1 to 3, that there is an upregulation of, or that there's expression induced if you co-culture them. And for CADM4, this is actually also significant. So this could hint at that there's a different, not necessarily CADM1, but another CADM-related interaction between mast cells and sensory neurons. And because we saw this increase of CGRP expression within the nerve bundles, we also looked at CGRP receptors. So the COLC receptor and the RAMP1 to 3 co-receptors. And here the same, the COLC receptor was not differentially expressed between the mono or co-cultured mast cells. But for the RAMP co-receptors, we do see that the expression was absent in the monocultures and started to be induced when we did co-culture. So that was still interesting. So if we then think about how these interactions could be happening, so for mast cells, most of the literature and also this study could hint at a relation between through CADM expression. So we could have CADM1 binding to CADM2, 3, or 4. So the strongest connections would be actually CADM1 to CADM2 or CADM3 to CADM4. And then in the fatal human asthma samples, we of course looked at this CADM1 expression, but as this was not very high in the in vitro model, we could not confirm this. And there was more suggestion of maybe a cadm-3, cadm-4 relation, but we didn't test this in the fetal human asthma samples. It's also possible that the nerves express other nectin-like molecules that could bind to cadm-1 on mast cells. And for eosinophils, the literature hints more at a eotaxin-1 expression on the nerve that then binds to CCR3, or expression released by the nerves to CCR3 on the eosinophils, or a connection, direct binding through ECAM1 on the nerve and LFA1 on the eosinophils, or VCAM1 on the nerve to VLA1. But we were not able to test this in an in vitro model specifically. So there were, of course, some limitation to the study that I just discussed. So we looked specifically at sensory markers using the CGRP. We were not able to study the cholinergic markers, and we were also unable to study any individual nerve fibers because of technical issues with the samples. Also, we had limited details on specific treatment of the asthma and of the phenotype of the asthma for most of the patients. So more in detail, only 5 out of the 12 individuals had a medical follow-up in the last 12 months before passing away, and it was unclear whether they had accessibility to inhaled corticosteroids for all of them. So that is good to note. And then for the in vitro study, we used HPC-derived sensory neurons in co-culture with mast cell line. So of course, this is a model of what would or could take place in the lung. And also there was no Th2 immune environment present in this co-culture model, which would be present in asthmatic lungs. Also, we were not able to perform the co-culture studies using an eosinophil related model, and we only looked at the changes in the transcriptome of the mast cell population, and we did not look at differences in the transcriptome of the sensory neuron population, or whether there were changes in functionality of the nerves or the mast cells. So overall, for this study, or for this presentation, I think it's important to note that neuroplasticity is a pathological feature of severe asthma, of which we still understand very little, and for which a lot more research is required to fully understand what is happening in the lungs of these severe asthmatic patients. We believe that this is the first documentation of this so-called neuroplasticity and these neuroimmune interactions in the lungs of fatal asthma patients. And we also quantified the CGRP expression that was increased in the nerves of these patients. We also saw that mast cell differentiation is induced in co-culture with HPC-derived sensory neurons, and together this hints at, sorry, the lights are going out in the office, that sensory nerve neuroplasticity and the changes in the bidirectional the communication between these sensory nerves and immune cells, they can contribute to exacerbations in asthmatic patients. And then I would like to specifically thank everyone that helped to the paper. Of course, Guilherme Dragunas, who was my co-author on this paper, and the rest of the team that helped with the publication, both at the University of Sao Paulo and the University of Groningen, the other people that supported the research, and of course the funding. And that was what I wanted to tell you about the presentation, and then I'm looking forward to the discussion.
Okay, thank you, Carly, for that very nice and exciting discussion and about presenting, the work that you did. It's a very reputed journal that you published your work in, so we are very happy to, have you share your work with us. But with that, I would like to open the conversation to our expert panelists. And for audience members, it's a reminder that if you have any questions, please use the Zoom chat function and enter the question as a message. And we'll get to those questions. So with this, I would like to invite Dr. Drake, Dr. Sue, and Dr. Taylor Clark to start your conversation and discussions, please. Thank you.
Let me start by saying what an impressive body of work you put together for this study, and I think it speaks to a number of features that we've been sort of kind of getting closer to understanding over the years. And of course, neuroplasticity being central to what we think is a mediator in fatal asthma, this excessive bronchoconstriction that in many ways is being driven by abnormal nerve function. Oh, I think we've got you.
I think so. Yes, I'm sorry about that. I was testing my microphone. Right. That was dreadful. Keep going. Sorry.
No. So let me start by just commending you for this really impressive study, super interesting data that you've generated, and I really like the fact that you've translated these findings in human tissues, looking at both eosinophils and mast cells in the relation to neurons, as well as then translating that into this stem cell culture model as well. So I think it's a really powerful thing that you've set up here. I'm curious, as I'm thinking about this, one of the questions that always comes up is a little bit of a chicken and egg question. You've described nicely how mast cells are changing after they arrive at neurons, and yet I guess the question that I start with is the mast cell that's arriving at a neuron, is it actually pre-programmed to go there? Is it something about that mast cell, either in the blood or the minute it engages tissue, that then sends homing signals to say, okay, I'm going to migrate towards a neuron? Or is it actually once it engages in the neuron that you're seeing a bunch of changes in the inflammatory cell patterns of expression? Do you have a sense of that? And it's probably an unknowable question at this point, but what are your thoughts?
Yeah, so thank you for your question, first of all. So we, of course, know that the mast cell fully differentiates once it arrives in the tissue. So from the blood, then it arrives in the lung, and then it matures. My take would be that it doesn't necessarily is pre-programmed to migrate to this lung. I think that, or to the nerve. I think the nerve expresses these markers, which other cells can also, that then result in this attraction, and that then it would change, would be my take, or that then due to the interaction with the nerve or the release of these neuropeptides, I don't even think specifically direct contact is required to induce this degranulation always. But it's of course difficult to see. It would be very interesting to do some spatial analysis or transcriptomics or proteomics to see if there's a difference between mast cells touching the nerves or not touching the nerves. But that, of course, still doesn't answer the question whether these mast cells that are touching were already pre-programmed. So that would be very interesting, and not specifically, I wouldn't really have one direct answer on how to find out if there's this pre-programmed mast cell that really migrates to the nerves, considering they already changed once they arrive in the lung. So it would really be a sort of a step-by-step analyzing these mast cells at different points, and sort of maybe in a murine model marking them and seeing if which ones do adhere to the nerves and which ones do not, would of course be very interesting. But my personal take would be that there's not specific mast cells that migrate, but that they sort of get activated due to this contact, but that's not a specific population per se.
Tom, you were weighing in there. Did you?
Yeah, no, I think that's a really hard question to answer, but I really liked your idea, Carly, of like a spatial transcriptomics kind of approach. That's exactly what spatial is trying to get at, the kind of the relationship between those cells which are touching and those cells which are just within the vicinity. And if you see a gradation of those kind of transcript signal changes that you saw in the in vitro culture, I think that would be a really cool thing to look at. I was struck by as you clearly said in the limitations, you're looking at nerve bundles here, and you are unable, unsurprisingly, human tissue, to study the nerve terminals themselves. And I thought I would bring into this conversation, the concept of, well, what could the sensory nerves be sensing at that bundle stage? I don't think it's really clear to to me. Maybe others have an idea, but I don't know where in the sensory nerve arbor, the terminal versus the axon, that it's actually a sensing nerve. There's been studies, we have done in obviously in laboratory animals that you put the classic stimuli of these sensory nerves, capsaicin or something like that, onto the nerve terminal, and they fire action potentials very vigorously. But if you put the capsaicin onto further along the nerve, that sensitivity decreases to the point where they're just not sensitive at all. And so my thoughts are that perhaps what you're seeing here is interactions. I mean, I don't know where we are in the anatomy at, for the images that you've got there. That's the big open question to me: is there actually a functional interaction between the sensory nerves that you've detected with your CGRP staining and the cells? But I would say that what is certain is that the immune cells can, instead of triggering the activation of the nerves, they could be changing their excitability, modulating through inflammatory processes, the voltage gated channels, which are responsible for not the initiation of the activation, but the translation of that sensitivity into actual action potential discharge. So I think that's something that we have no idea about. And I think your work really shows that it's not just the interaction at the nerve terminal, but also along the axon, maybe two different compartments that needs to be considered.
Yuzhuan, you've done a lot of work looking at various segments of axons in relation to airway and neurobiology. What are your thoughts?
Yeah. Thank you for including me in this exciting discussion and hear this important question. So I see from your presentation, this is really an open question, but you give a lot of human data suggesting that by quantification, there are increased nerves, terminals, there are increased muscle cells, so indicating fundamentally, there may be an increase of the functional connection between both of them. So I think this is really exciting. And I think your method, like how you quantify the nerve increase, how you quantify the increase of the immunocell should be highlighted as well, because we know that there are lots of variations, like case to case from different disease models. So I think set up a model like to quantify the increased density of the neural innervation and the immuno is also very important for us to study. So I also want to follow Dr. Tom Taylor's question about CGRP. So I see you have an increased density of CGRP positive nerves, which you also include a possibility it may be an indication of the nociceptor phenotype change in addition to the nerve density increase. Can you talk a little bit more about that thought, like how you think about the nociceptive pathway will contribute to this?
Thank you.
Thank you.
For your question. Yeah, so we started off, of course, with the PGP quantification, and then we saw this increase, and because we specifically were interested in sensory nerves, we looked at the CGRP expression, and we saw that it is increased in the nerve bundle, or if you look at the specific nerve bundle, they have this higher expression of CGRP, but if you then correct for the overall increase already present for these nerve bundles, then it sort of disappears. So that would indicate that you really see in these nerve bundles that the CGRP is increased, which is of course very interesting, whether that's then a nociceptive change or whether the bundle sort of is more sensitive, because we also of course link it to this hypersensitivity maybe due to this, there's lots of theories I could think of why the CGRP could potentially be increased. Is it because they are more sensitive because of these nerve cells or immune cells in the vicinity? Is the increased CGRP a reason why there's this increase of immune cells, that they're more attracted to the nerve bundles? I think that's a very interesting question to sort of also maybe chicken and egg, like what is the basis for the other? I think there's not yet a clear answer on this. And then
are you able to see any CGRP positive neurons or cells within the lung besides the nerve terminal?
I think the PNECs also express so express CGRP. So I think in the study we were able to differentiate between the CGRP and the PNECs and the bundles really, but they do express also CGRP. So it's not only the nerve bundles that express the CGRP.
Sorry,
and what the effect?
Can you clarify what you mean by PNECs?
The neuroendocrine.
Right. The PECs, right? Yeah.
PNECs. The what is it for neuroendocrine cells?
It's pulmonary neuroendocrine cells, you mean?
Yes.
Okay.
And then what the effect of this increased CGRP? I think it's still speculation at this point what that means. Whether they have been more activated, whether it's stress because of the fatality of the asthma attack. That's of course also, I mean, that could already induce maybe the CGRP increase. I think there's not a clear answer yet on why we see this increase. I think this study really sort of identifies this increase, and we sort of linked it to the presence of the mast cells to investigate, is there a link there? But I think that's not the only interesting avenue that you could pursue from the fact that the CGRP is increased in these nerve bundles.
Well, the really interesting thing about these big nerve bundles is, I mean, we've been focusing on the sensory side, and your data suggests there's changes on that pathway. Our group's been really interested in the parasympathetic, the cholinergic innervation there as well, and we know that those axons are both present in those bundles, and they release, particularly things like eotaxin to drive eosinophil migration, which suggests that both limbs that you referenced at the start of this are actively, seeking interactions with immune cells. And one of the questions that always comes up is why? Why in the world would a neuron want an immune cell to arrive at it, to physically interact with it, and to release a bunch of mediators? Do you have thoughts as to why we've developed this physiology and this neuroimmune interaction?
Why they would attract? Good question. I think the most, for me, one of the interesting things is which one is the start. Is the innervation increased, and does it attract the immune cells? Is the immune reaction that we of course know from asthma, is that what sort of induces this increase of nerve growth and outgrowth, and do they grow towards these immune cells? I think that's still a question that in my head that I'm not sure of which way around, or is it bidirectional? Do they increase each other? And then indeed the question is why. I mean, we know now, or mechanistically we can say that both of them secrete either neuropeptides or immune modulators that attract the other. Is it because they want to increase, for immune cells, for example, they want to increase it's sort of a feedback loop, positive feedback loop? Do they want to sort of get this immune reaction going even more? You of course see often positive feedback loops in the Th2 immune response. Do they use these nerves for that, to sort of increase their own presence? For nerves, is it simply that they also want to grow more? I mean, or is there really something that they can get from these immune cells? That would be, I think, something to investigate further. I don't think I have a direct answer there.
There are a couple of questions in the chat.
Yeah, I think Tu, can you ask the questions to Carly, and then we go from there?
Yeah.
So
So
Carly, in the chat, Melissa had asked, how could the identification of the clinical markers of neuroplasticity and neuroimmune activation guide personalized prevention and treatment strategies in patients at risk of fatal asthma?
So it's a good question. Of course, in the end, we want to implicate it in a clinical setting. I think really the measuring of these nerve markers is quite difficult and quite invasive, would be for really asthmatic patients. Then I think it might be easier to measure, for example, these nerve or neuropeptides that are secreted by immune cells and see if that could be an indication. I think now at the moment the field is not far along enough to really identify already clinical markers that we could measure to really see who's at risk. It would be interesting to sort of follow the development and see if there's an indication early on already that people that in the end develop this persistent and severe asthma early on already have an increase of neuroactivity or nerves present, or they have, you can sort of differentiate them on a clinical marker who is going to develop this and who is not. But I don't think at the moment there is one specific marker that we could identify for this, because we're not sure which immune cells specifically play a role. We look here at mast cells. There's a lot of studies on eosinophils, ILC2s. So I think there's a broad scala of cells involved in this feature that we would first need to really research mechanistically what is happening to identify markers that we could then use and maybe prevent this in patients. And for prevention, if the base, that's also the chicken and egg question, I guess. If you are not sure if the nerves are activating the immune cells or the immune cells activating the nerves, where do you start preventing? Do you prevent the binding of these neurotrophins and neuropeptides to the nerves, or do you prevent the further activation of these immune cells? I think that is something that would first need to be investigated further to really see which of the two, where you can sort of interfere in this bidirectional communication to see if that changes the eventual clinical outcome. If that answers the question.
I think you've identified something there, Carly. You know, the neurotrophins, I believe that there was a number of studies in the late 90s, early 2000s, where they were able to detect nerve growth factor and maybe also BDNF inside BALF fluid.
Yeah.
So that could be, I mean, obviously it's an indirect marker, but you would think that nerve growth factor and BDNF would be two of the most likely contributors to the neuroplasticity, at least the sensory neuroplasticity. I can't speak for the efferent side of things. I think that's plausible that could be measured, but I don't think I'm not quite sure that's ever been extrapolated to various asthma phenotypes, but maybe it has. I'm not aware of that.
No, you're totally right. There was a lot of work looking at neurotrophins, and we, in many ways, this predated a lot of the phenotyping discussions.
Yeah.
So this was the era right when we were just starting to think about anti-IL-5 biologics for asthma, and animal data was suggesting this was going to work, and those early anti-IL-5 studies in humans didn't. And it was really the birth of the concept of phenotyping and how we should be stratifying. When I think about the neurotrophin story, I think there may be a lot to learn from the phenotyping story with anti-IL-5s, and that we kind of moved on from the field because we weren't quite sure which patients this was relevant to. And yet there may very much be a neurotrophic signature in certain patients. When we think about the role of neural dysfunction, and particularly how it pertains to airway hyperreactivity, how that subsequently would map to things like fatal asthma, it does make me very interested to understand whether people who are at risk for more severe exacerbations or even fatal asthma may have particular markers that we should be paying attention to? I think it's a great question that we should try and answer as a field.
Yeah, adding to that point, I think the in vitro culture model may be a way like trying to talk about the personality, even the treatment. Like your culture model, you know, we culture the neurons for a long time, and we can add in a bunch of, you know, antagonists or even blockers that we are interested. And those neurons are derived from patients, so they have their unique asthma status. So I think that is maybe a way like we're trying to think about, can we use like in vitro culture personnel, the treatment option, and then test the efficacy in something. So.
Yeah, the real power in the iPSC kind of approach is that you can take the individual's genetics into account.
Yeah.
But I'm not convinced that I'm sure epidemiologists would cry at me saying this, but I'm not entirely convinced that there's much of a strong genetic component to these fatal asthma situations. We know that there's a genetic component in the probability that you'll be diagnosed with asthma, but that component is pretty small compared to, say, whether or not you had a viral infection when you were young or something like that. So, but I certainly agree that the human stem cell derived nerve cell approach is essential for us gaining more insights, because of course we just can't get live neurons from the vagal ganglia. I did want to actually address one thing about these nerve cells that, Carly, maybe you could clarify for me. You said that they were vagal neurocrest cells, but the majority of vagal neurons are placodally derived, i.e. the nodose neurons are from the placodes, whereas the jugular neurons are from the neural crest, and the jugular neurons are fewer than the nodose neurons. Can you explain why you took that approach? Was that just a convenience thing, or you were targeting the jugular neurons for a particular reason?
So very honest answer, we already had the protocol for the vagal neurocrest precursor model, from which we derived cholinergic neurons prior, and then I alterated the differentiation protocol to generate a more sensory phenotype. So that's why we are using these cells, because yeah, the protocol for the vagal neurocrest precursors was already present. But it's a good point that there's a differentiation between the nodose and the jugular derived sensory neurons, indeed, and that we for the model we did not take that into account, basically.
Yeah. I mean, jugular neurons have been identified as being very important for cough, and a group at I think in Georgia have said that they think that they're very important for the bronchospasm, neural airway hyperactivity. So I'm certainly not suggesting that these were not important nerves to look at, but very, very interesting.
Thank you.
Ben,
And I think there's
to ask another question. Is there any evidence that the sensory nerves adopt a chronic neuropathic state in asthma?
So for this study in the in vitro model, unfortunately we were not able to look at the sensory population of the in vitro model, and we only were able to look at the transcriptome of the mast cell population. And I think in human samples, we have not really, or there's not a lot of data on transcriptome of the nerves itself. There have been people isolating, for example, the DRGs, but that's only a minor population of the lung innervating neurons. And if you isolate, for example, the vagal ganglia or the cell bodies thereof, then you also look at nerves innervating other portions of the body. Like, it's not only the lungs that are innervated through this nerve, through the vagal nerve. So I think that really limits what we can look at there. So from what I know, there's no direct proof, and in the stem cell culture, so that's because I see that's the second part of the question, is we were not able to look at the transcriptome of the neuronal population.
I also had a second question, but I'm asking him to clarify just because I could have missed part of what he wanted to ask. So Sven, if you're available, please ask the question. But in the meantime, I also have a question myself that's more basic level. Is that your histopathology is based on fatal patients with fatal asthma? And I was just wondering, is it patients that, how did you diagnose them? Did they come in with ARDS in the ICU? They had a history of asthma prior. They just ended up not becoming unresponsive to steroids and bronchodilators. And then also, do you feel like your current thoughts on how the pathology of this is playing out with your mast cells and immune response is similar in patients with fatal asthma versus even in an acute state or chronic state?
Thank you. So for the patients from the study, so this was a collaboration with the University of Sao Paulo. So I was not directly involved in the collection of those samples, but so I hope I'm not saying anything wrong, but so they were collected over the years in Brazil, and at that point in time, so I think between 2004 and 2009, they were pathologists confirmed at least the asthma pathology, and in Brazil at that time there was not adequate access to oral corticosteroids, so that's why they were not at least monitored on this treatment. So they had other treatments, but it was not available at that time in Brazil. So that's why it's sort of a unique cohort of patients. And I think the confirmation of the asthma pathology was already done after. So some of these sample collections were even older. And that is the first author, Thais Maat, on the paper, was involved in collection or collection of the samples, but she really was the one who collected the cohort data. But what that is, as far as I know, why this model or this cohort was used. So, and why they didn't have access to the inhaled corticosteroid, but were sometimes treated with other asthma medication. And then your second part of the question, I forgot. I'm sorry.
It's okay. I was just wondering first about the patient cohort. Like, were these patients that came in with an acute asthma attack and died, and therefore you based your information off of that? And then if so, then based off of that, do you feel like your immune response, like the mast cells, vary in their response in like a fatal asthma attack versus an acute versus chronic response? Or do you feel like the mast cells pretty much overall respond the same way, but maybe to just a varying degree?
Mm-hmm. Yeah, so for the I would say yes to the question about the fatal asthma attack. Is they were brought in and there were no underlying pathologies. I think that's why also it was identified as fatal asthma attack. And then for the mast cells, so this is I think part of the chicken and egg question. So I think that my take on it is that the mast cells and the nerves sort of in a bidirectional communication activate each other. So I would from that suggest yes, that mast cells maybe become more activated through this either cadm1 interaction with these nerve cells or through the release of these neuropeptides. Whether there's really, except a few papers, direct proof of this, I think is difficult to say, but that would be my hypothesis on this. So then I would suggest yes in severe asthma, because this is where we observe the neuroplasticity. You could say that there's this increased role for the mast cell, and I think also other immune cells. It's not just the mast cell, but also the eosinophils and these ILC2s, and that this can contribute to these exacerbations. But I don't think the full direct proof for this has been. I don't think we have that yet to really confirm and really prove and say, okay, there's a higher contribution of these immune cells due to this neuroplasticity. But I think that is the hypothesis that I would say now to further investigate whether this is actually true.
Yeah, I'd say, we come up against this nearly always in this field, which is what is the evidence that nerves are contributing both to how the patient feels and also the extent of the immune responses. And I think this is something which is a real problem for the researchers in this field, because I think we fight against a lot of thought that the nerves are not important because we haven't yet shown that they are. It's a circular argument. But I think if we just think about this from the very basic concept that that we know that people, that many asthmatics feel bad even when their immune responses are quite well controlled by the corticosteroid treatments. But they still feel bad. They still have, in some cases, a very active cough reflex. And they have what appears to be an airway hyperreactivity, which a lot of research suggests that there is a neuronal component. But in humans, we haven't got a drug yet which actually targets the nerves themselves. And until we have a drug which can target the nerves, then it's very hard to find any actual direct proof, other than it is self-evident that people cough more and they have more reflex bronchospasm in asthmatic individuals. I don't think we've proceeded in 30 years. We have not really made a critical finding in 30 years in human asthma. But in the animal models, I think we've made big strides in the last 15, 20 years. And so it's real. So it's your work is really great, Carly, because it shows that there are more and more stuff that we can get from humans. But I worry that we still are still saying the same thing that we would have said 20 years ago, which is we don't have any direct proof in humans.
That represents the great opportunity, right?
Yeah. It's a good problem to have.
Yeah.
Yeah, I think on that note, we are to the hour right now. So I'd like to thank our speaker, Carly, as well as all of the experts, for your valuable time and participation to share your knowledge with all of us here today. Also to all of those who joined us and submitted questions and, truly made this Journal Club webinar an engaging discussion. Like the experts said, this will be an ongoing discussion and conversation that we are all committed to for the rest of our research life. So on that thought, I think I would like to thank everyone at the ATS, the ATS RSF, the organizing committee, for coordinating all of our efforts and making this webinar a success. If anybody has any questions or suggestions for any future topics, speakers or presenters, please feel free to contact your ATS Assembly web representative, and we can converse then. So with that, I'll conclude our Journal Club webinar for today. Have a great rest of your day. Thank you, everyone.
Thanks, everybody.
Thank you, everyone.
Thank you.